Okay, good afternoon. Welcome back. Today we're going to talk about intrinsic and innate defenses, but first, a little update on SARS-CoV-2 and COVID-19. This is a very interesting experiment that is in a manuscript submitted to the New England Journal of Medicine. It is coming from labs at NIH and Princeton and UCLA. And what they did is ask how long does the virus remain infectious in different things, aerosols and different surfaces. And they compared SARS-CoV-2 with SARS-CoV-1. And so we have half-life and detection limit. These are all done by infectivity assays. So, for example, in aerosols, and they make these in the lab, they can generate aerosols and collect them and see how long the virus lasts in them. SARS-CoV-2 and 1, both about 3 hours in aerosols. And copper, 3 hours. That's half-life. Cardboard, 8 hours for SARS-CoV-2, 2 for SARS-CoV-1. Steel and plastic a little longer. So these are common surfaces. So that's the half-life. In other words, every 8 hours on cardboard, you lose half of the infectivity. Now, how much you need to get infected, we don't know. But all this tells you is that the virus can last a good amount of time on surfaces. Copper is known to be antiviral and antibacterial, although it's not much use because unless you're going to wear copper sheathing, it's not going to help you too much. Then we have detection limit, which is interesting. And how long can you detect infectious virus in aerosols and so forth? And you can see a couple of hours on aerosols, copper, a little longer on cardboard, steel, and plastic. Similar numbers for SARS-CoV-1. Okay, so this tells you that the virus is stable in aerosols and on certain surfaces. So it could be transmitted by aerosols, which we know is happening, and also by fomites, by contamination. So that's a really important experiment. But remember, this is laboratory conditions. This is a controlled temperature and humidity. And in the real world, it's going to be different. And it could be longer or shorter, depending on that. And also remember, if the air is circulating, the aerosols can go farther. Because we say about 2 meters is as far as the droplets are going to go. But if the air is circulating, it'll go longer. Someone just sent me an article saying, in this bus somewhere, people throughout the whole bus got infected. Well, they probably have a circulating system, just like the cruise ship, where they circulated the air throughout the ship and they spread virus to everyone. So, these parameters are laboratory determined. And one more thing I want to tell you. I read a good article today in Medium. And he's saying basically what I've been saying for a while is that the case fatality ratio, not rate, he's saying it's not rate, but you know that already. So let's say ratio. Varies according to the country, 0.5% in South Korea, 0.9% outside of Wuhan, right? Higher in Wuhan. And I've always said this, this depends on age and also the healthcare. And he's agreeing with that. Countries that are overwhelmed, in other words, the healthcare system, if the system is overwhelmed, they will have a higher case fatality ratio. If you're prepared, like South Korea and the rest of China was, it'll be lower. And so Italy is higher, Iran is higher because they're not prepared. And now we're getting into the phase of this outbreak where preparedness matters because the virus is here. It's going to increase exponentially all over the U.S. We're going to have a lot of cases. And the real key is whether the healthcare system is overwhelmed. And that's what preparedness is all about, having enough hospital beds, knowing how to treat people, and so forth. I don't know if we're prepared. I think, if 1% of the infections required hospitalization, that could easily overwhelm the system, given our population. And the other point here is that we only have 1% of the masks we need to cover the needs of our healthcare workers. We have only 12 million N95s and 30 million surgical masks in stock, and we'd need about 3.5 billion. So we could exhaust these in a couple of weeks. I think that wearing a mask can help, but the CDC is not recommending it. And I think that's because they realize there's not enough to go around. And also because people don't use them properly. If you don't use them properly, they don't help. So 2 interesting points there. Okay. Let's talk about intrinsic and innate defenses. Last time we talked about infection basics, and I said we'd come back to infection. First, we have to learn a little bit of immunology, not just from the perspective of how it protects you from infection, but also how the immune response actually causes disease. So this is how I view the host defenses as a series of barriers over which viruses must overcome to establish an infection. So first we have anatomical and chemical barriers that are always present in us. Things like mucus, saliva, stomach acid, tears, your skin, a physical barrier, the outer layer being dead, even scabs. And then there are proteins that are produced by the bacteria in your skin called defensins, and they're actually, some of those have antiviral activity. So these are barriers that are always present. They're very effective. They can stop a lot of viruses. If viruses get past those, then we have our intrinsic defenses. And these are ready for immediate deployment. They include interferons, autophagy, apoptosis, microRNAs, CRISPRs, and others, and we'll talk about some examples. If that doesn't work, you have innate immunity, and this requires minutes to hours to deploy. It includes natural killer cells, complement, antigen-presenting cells, neutrophils, and cytokines. And then finally, after a couple of weeks, 2 weeks or so, the acquired immune response can kick in, or the adaptive response, which comprises T and B cells, and we'll talk about that next time. That's tailored to the pathogen. Today we're going to talk about intrinsic and innate immunity. So let's define them. Intrinsic defense is always present in the uninfected cell, ready to go. The runner is ready to go there whenever a virus is impinging on it, and these are some examples. We'll cover some of them today. And the innate immune system is slightly different. It's induced by infection. It's not always on, and you could look at the innate system as you need to warm up first, minutes to hours. It's pretty quick, but it still has to be turned on. And then the adaptive, which, again, we'll talk about next time, it's tailored to the pathogen. You make antibodies and T cells that recognize the pathogen specifically, so they take about 2 weeks to kick in. So let's look at some examples of intrinsic defenses. One is RNA interference, and this comprises, say, here's a virus, an RNA virus infecting a cell. The RNA is chopped up by a cell protein called dicer into short RNAs, typically 21 bases long. The strands are separated. They are placed in association with a RISC complex, which contains another nuclease called AGO or Argonaut. And then this siRNA, which is what it's called, originally derived from the infecting virus, that is used to target the viral RNA. So if this virus is replicating, there's viral RNAs being made, this siRNA will base pair with the RNA, and then the Argonaut will chop it up. So it's an RNA-based defense, and it's specific for the virus because it's based on base pairing. Now, where does this work? Clearly in plants and in invertebrate cells, this is an antiviral defense for sure. However, there's a lot of controversy over whether this is present in mammals as an antiviral defense. We do make microRNAs, as we mentioned before. The siRNA machinery works. If you put siRNAs into cells, say to knock down an mRNA, they will work. So the machinery is there, Argonaut and the RISC is all there. But we don't think that it's active as an antiviral defense, at least not in somatic cells. It might be in germ cells, but there's only been a couple of reports on that, and it's controversial. So it's clearly active in plants and invertebrates. We don't know about mammals. Now, with everything I'm going to tell you today, there's a countermeasure. There has to be, because these are such good defenses that viruses wouldn't exist unless there were countermeasures. And some countermeasures include binding RNAs. Viruses would make proteins that bind these RNAs and prevent them from targeting their genome. Here's another example of intrinsic defense. It's a family of proteins called the apobecs, and this is one example, apobec3, and this is what it stands for. This is a cytidine deaminase, which essentially ends up causing mutations in nucleic acids. And here we're going to look at what it does to HIV-1, a retrovirus. Now, normally HIV-1, here we have the particle on the left budding out of the cell. New particle infecting another cell, reverse transcription, you make DNA, of course, and go in the nucleus. All is good. And it's good because there's a viral protein made called VIF. And VIF recruits apobec3G, this particular brown protein here. It binds it, and it causes it to be ubiquitinated. And this whole series of proteins here are part of the ubiquitination machinery. So VIF, the viral protein, causes degradation of apobec3G. It sends the protein to the proteasome where it's chopped up so it can't get in the virus particle. And that prevents apobec3 from mutating the genome. Now, if you take out VIF from the genome, you make VIF minus viruses. Then apobec is not degraded. It's incorporated into the budding virus particle, the little brown ovals there. And then when they infect a new cell, the apobec goes to work. It mutagenizes. So when the RNA is present, it's copied to form DNA. And so we're looking at a GG base pair, two GG bases, just as an example. So we have RNA copied to a DNA, and then this DNA is mutated. This C is mutated to U, for example. And then when the DNA is copied, U now templates to base pair with an A, and you have a mutation. So you deaminate the C, you make a U out of it, and that causes mutations. And basically this happens throughout the genome, and it mutagenizes the virus so it's no longer infectious. So that's how this is an antiviral factor. And if it weren't for VIF, HIV wouldn't get anywhere, because apobec, again, is in cells. And it's an antiviral defense. And these apobec genes, on the right here is a diagram of all the apobec genes that are present in different mammalian species. And each line is a different apobec gene. And here's apobec3G. So we have 2 of them in us, but we have lots of other apobecs. And in different species, you can see they've all been duplicated. And we think this duplication is in response to infection. Over the many, many years of evolution, viruses have been infecting us, and we duplicate our apobecs to make more antiviral defense. And you notice here, who has the most apobecs? Bats. Look at all they have, probably because they have so many viruses in them, more viruses than any other mammal on the planet. And they have all probably arisen in response to the pressure of all those viruses infecting them, so they're being protected by this. So I think that's pretty neat to think about that. So that's an intrinsic defense, and there's the counter right there, VIF, for this virus. And other viruses have different ways of countering this, because this will inhibit both RNA and DNA viruses. Another intrinsic defense is epigenetic silencing. When DNA comes in the nucleus, it is modified so it becomes compacted. DNA, of course, is wrapped around nucleosomes, which are made of chromatin, and the DNA is wrapped around them. And when this entire chromatin is compacted, it can't be transcribed because it's physically inaccessible. But things like acetylation of histones can open up the DNA and make it active. Well, when foreign DNA comes in the nucleus, the cell silences it by modifying it in ways that cause it to be compacted, taking off acetyls, for example. But many DNAs come in the nucleus naked or with viral proteins on them. They're not chromatinized. And then the cell rapidly coats them with histones and silences them. And that happens in these discrete foci in the cell nucleus called PML bodies. So all these green dots are areas. These are virus-infected cells. These are the nuclei you see in blue. And the cell makes these foci, and it puts the viral DNA in there and silences it. And of course, viruses have to encode countermeasures, DNA viruses in particular. Otherwise, they wouldn't replicate. And so for example, the human cytomegalovirus genome encodes a protein that it degrades a protein, DAX, that's needed for histone deacetylation. Deacetylation would compact the chromatin, so it keeps it open. And Epstein-Barr virus, adenovirus proteins affect the formation of these PML bodies. And then finally, retroviruses. When retroviral DNA comes in the nucleus, it's immediately chromatinized and silenced. And the only way it gets expressed is to integrate. The actual integration, and often it's in active chromatin in the chromosome. That integration act desilences it and allows it to be expressed. So it's not really a protein that counters epigenetic silencing. It's the physical act of integrating that does that. Apoptosis is another intrinsic defense. When cells are infected or stressed in a variety of ways, they undergo this process in which they kill themselves and they degrade the chromosomes and they produce apoptotic bodies, which are pieces of the original cell. And the idea is that they will be taken up into other cells and reused. This cell is going to die, the apoptotic cell. And here's an actual photograph of cells blebbing off these apoptotic bodies. But important for us is that, of course, this is a defense against infection, but this process is monitored by what we call sentinel cells, like macrophages. They will take up and phagocytose apoptotic bodies and will look for foreign proteins and nucleic acids. And if they are foreign, like virus-infected proteins, then they will initiate a response. And we'll take a look at that later today. Apoptosis is highly regulated, both within cells and by viral proteins. And there are two ways that you can start the apoptotic program in a cell. There's what's called the extrinsic pathway, where some ligand binds to a cell surface receptor. So here are 3 different cell surface receptors that bind different ligands: Fas ligand, trail, tumor necrosis factor. And we'll come back to Fas ligand later in terms of immunopathology. But the binding of these ligands causes cleavage of caspases, procaspases, to caspases, which eventually induce apoptosis in the cell. So that's the extrinsic pathway. There's an intrinsic pathway when there's mitochondrial damage in the cell. The mitochondria release cytochrome C, which stimulate the formation of caspases that induce apoptosis. And on this slide, you can see that viruses both stimulate and inhibit apoptosis. Viruses would stimulate apoptosis so that maybe the cells would release virus in a better way, and they could stimulate at the extrinsic or the intrinsic levels. And they also inhibit apoptosis. These viruses encode proteins that inhibit signaling through the TNF receptor, and as do these, and some inhibit the intrinsic pathway as well. So again, regulated, because if a virus infects a cell and the cell kills itself, the virus isn't going to be able to reproduce, so they have to inhibit apoptosis. There's also, of course, a really ancient intrinsic defense in archaea and bacteria, which is, of course, called CRISPR. Clustered, regularly interspaced, short palindromic repeats. This was originally discovered as a defense, an intrinsic defense in microbes. Of course, it's now been repurposed to do gene editing, but it is present in these bacteria to protect them against infection. The way it works is a phage infects a cell. The genome is chopped up and integrated into the host DNA in these long arrays. That's where the short palindromic repeat comes from. And then this array is continuously transcribed into CRISPR RNAs. And so if this cell happens to survive, and a fraction of infected cells will, then the next time they're infected, these CRISPR RNAs, together with the Cas nucleases, are going to degrade the incoming viral genome, as shown at the bottom here. So that's another defense. It's always present because these CRISPR RNAs are always made in archaea and bacteria. Intrinsic defenses are always present. Which of the following are included? Antibodies, T cells, epigenetic silencing, skin, and mucus. All right, let's see how we did. Epigenetic silencing. Exactly. That's always present. Antibodies take 2 weeks. T cells take 2 weeks. Those are adaptive. These are not intrinsic. We called those physical and chemical defenses. Okay. So it's a little semantic difference, but I want you to understand that these are different from physical defenses. They involve proteins and nucleic acids, for example. So let's talk about the innate immune system, the other part of today. That's the second wall of bricks there, activated within minutes to hours. We're going to talk about how it's activated today. And it comprises cytokines, sentinel cells, and among those are dendritic cells, macrophages, and NK, or natural killer cells, and complement. So this has a role in stopping infection, as you will see. But another key role is to inform the adaptive response when infection gets to a certain point and it can't be handled by the innate response any longer. So they're talking to each other, and how exactly they talk to each other we're going to look at today. So let's talk first, how was this discovered? How does the innate immune system recognize microbes and not self? And this was a big problem because we knew for many years that there were these innate immune components, but we didn't know how they were sensed. So this story begins in 1980, not too long ago. 2 scientists in Germany were studying fruit flies. They were not interested in infection. They were developmental biologists, and they started, and it may sound weird today, but they started mutating genes in the fly and seeing their effects on development. It's something that people do all the time now, but at the time it was novel. And they identified a gene that was important for establishing dorsal ventral axis in the embryos. And they called it the toll gene, for which they got the Nobel Prize in 1995 for finding a gene that controlled development. And why is it a toll gene? Well, this little German phrase here, das war ja toll, supposedly is why Nusslein Volhard called it that, because when they saw the phenotype, she said, wow, far out, which I guess is this in German. Later, it was found to have also a role in immunity of flies itself. And then in 1997, they were identified in mammals, and they're called toll-like receptors. So the original gene is the toll gene. And then looking for orthologs in mammals, they found toll-like receptors. And these now have turned out to be really important for innate defenses. And they're toll-like receptors are part of a whole family of cell proteins that are what we call pattern recognition receptors, PRRs. And there are toll-like receptors, C-type lectin receptors, CLRs, nucleotide binding oligomerization domain-like receptors, NLRs, and finally RIG-I-like receptors. And these are in different places in the cell. CLRs are transmembrane, plasma membrane proteins that recognize sugars from fungi and bacteria, so we won't talk about them anymore. Our nucleotide binding, or NLRs, are cytoplasmic sensors, like this one here in the cell, that recognize bacterial, viral, parasitic, and fungal products. We call these pathogen-associated molecular patterns, PAMPs, something that is specific to the pathogen or the microbe and not to us. We won't talk much about those in this course, but we'll focus on toll-like receptors, the plasma membrane proteins, or they're also in the endosome. They can be in both places. They recognize proteins, nucleic acids, and sugars. And the RIG-I-like receptors, which are cytosolic sensors of RNA, and we'll also look at DNA sensors as well. So here is a little bit of information on how these molecules recognize pathogen-associated molecular patterns. Here on the left, we have 2 plasma membrane toll-like receptors, 2 and 4, which recognize viral proteins as being foreign. And when they bind the viral proteins, it initiates a signaling pathway that ends up in the synthesis of cytokines, including interferons, which we'll talk about in a bit. We also have endosomal toll-like receptors that recognize RNA and DNA, TLR3, 7, 8, and 9. They recognize different things in the endosome, which is where the viruses are coming through, of course. And so double-stranded RNA, single-stranded RNA, and DNA, CpG of DNA. And again, when these nucleic acids bind the TLRs, it initiates a signaling cascade. In these cases, it allows a transcription factor, either IRF37 or NF-kappa B, to get in the nucleus and turn on the synthesis of interferons. And so this is foreign. RNA of these types are recognized as foreign. You'll see why in the next picture here. And then on the surface, when viruses are binding these TLRs, it's foreign. Here on the right is a detail of the pathway through the cytosolic RIG-I-like helicases, RIG-I and MDA5. These are two cytosolic proteins that recognize nucleic acids. And again, the TLRs are typically, the ones that bind nucleic acids are typically endosomal. Here, RIG-I will recognize a double-stranded RNA with 5' phosphates, triphosphates, short double-strand RNA without phosphate, and single-stranded RNA with triphosphate. These are all things that the cell doesn't make. The cell doesn't make triphosphate-ended RNAs. They're all capped, or if not, they're in the nucleus. So these are recognized as foreign. They activate RIG-I, and then MDA5 recognizes long double-stranded RNA, which again is a viral product. So that's why these are specific for foreign PAMPs, if you will. In both cases, when they bind a nucleic acid, they initiate a signaling pathway, which goes through this mitochondrial protein and eventually activates protein kinases that phosphorylate transcription factors, IRF3, 7, NF-kappa B. In the case of 3 and 7, they dimerize, they go in the nucleus, turn on transcription of interferon genes, and NF-kappa B. Phosphorylation of this inhibitory subunit releases it so it can go in the nucleus and again bind at the promoter and turn on the synthesis of interferon. So that's how these work. That's how they recognize foreign PAMPs. And there's also a signaling pathway for sensing DNA. So DNA viruses infect cells. They're taken up typically by fusion at the membrane or by endocytosis. And often viral DNA leaks out into the cytoplasm during this process. And the cell does not want DNA in the cytoplasm. Even if its own DNA ends up there somehow by nuclear leakage, it's going to stimulate this pathway. And so what happens here, there's a protein in the cytosol called cGAS, cyclic GAMP synthase. So this is an enzyme that recognizes DNA, and what it will do is take ATP and GTP and make a cyclic molecule out of it, cyclic GAMP. And the structure of that is shown here on the right, really interesting molecule. So you have a GMP and an AMP, and then they're joined via their phosphates to make a cyclic molecule. So that molecule is produced by cGAS when it binds DNA. And then cGAMP binds to a endoplasmic reticulum protein called STING. And rumor has it that the person who named this was a fan of the police, the musical group, not the law enforcers. And that binding then pushes STING through the Golgi. It activates two kinases to phosphorylate transcription factors. Again, IRF3 and NF-kappa B. They go in the nucleus and turn on the transcription of cytokines. So again, it's very much like the cytosolic RNA detecting proteins, RIG-I and MDA7, except this is for DNA. Of course, viruses encode proteins that modulate sensing. And I don't actually expect you to memorize any of this. I just want you to know that there are antagonists at every step. So here on the left, we have cytosolic sensing of RNA virus genomes by MDA5 or RIG-I. Just one example. Kaposi sarcoma herpes virus protein blocks the ubiquitination of RIG-I, which is normally needed for it to start signaling. And so that inhibits the pathway. The viral polymerase of hepatitis B virus blocks RIG-I-mediated signaling. And you could see many more interventions by all these red bars. Hepatitis B, let's say hepatitis C protein, hepatitis C virus encodes a protease that actually cleaves MAVs. So it doesn't work. And all these other viruses are intervening. Here are some ones that are interfering with DNA signaling, sensing and signaling. So the effect of these inhibitors is to block the production of interferon when viral nucleic acid is sensed by the cell. And that's important because if interferon is made, it's a powerful antiviral, as you will see. Which of the following allow the innate immune system to distinguish microbes from self? Cytoplasmic helicases and toll-like receptors, antibodies, apoptosis, apobec, all of the above. How did we do? Cytoplasmic helicases and toll-like receptors. That's the answer. So which of the following allow the innate immune system to distinguish microbes from self? That's it. The others do not. The others are defenses of some sort, but they don't actually allow you to distinguish. These helicases, as I've said, are the ones that do that. So far, we're on the innate defenses. The various sensors have sensed an infection. What happens next? Well, what happens is the production of cytokines, which are cellular proteins, typically soluble, involved in innate defenses, and a big number of cytokines are the interferons. These are discovered in 1957. A number of investigators had taken chicken cells. They had inactivated influenza virus and just put it on the cells, and the supernatant of the cells had something in it that if they put it on another cell and infected it would inhibit infection. So they call it interferon for that property. And many years we had no idea how it worked. And I'm going to tell you how they work today. But these are basically made by virus-infected cells. They can also be made by uninfected sentinels. So in the virus-infected cells, the infection induces the interferons by sensing of nucleic acids, as we just talked about. In an uninfected cell, the sentinel can make interferon in response to products that are released from infected cells, like nucleic acids that are sensed. You'll see these sentinel cells have sensors, and they'll make more interferon. So here's an example. We have an epithelial cell, could be in respiratory or GI tract or elsewhere. We have a layer of epithelial cells. They're getting infected. The infection is sensed. The cells produce cytokines, which are the red dots here. And they're soluble. They diffuse out of the cell. And they will attract other cells to the infected area. And so here's an immature dendritic cell, which has come in. It's sensed these cytokines. They're always patrolling our bodies, these dendritic cells. And when they sense an infection by virtue of cytokines, they come in. They're activated by the cytokines. They can also pick up pieces of dead cells. If these cells die and release viral proteins and nucleic acids, the sentinels can pick them up and respond further. And then at some point, they go into the lymph node. They actually mature. They become mature dendritic cells. They will go in the lymph node and then have a conversation with lymphocytes there, which we will talk about next time. Because that's the link between the innate and the adaptive response. These dendritic cells are going to say, okay, there's an infection here. We can tell by the cytokines and the things being released from infected cells. Now let's go talk to the adaptive response. So there are three major types of interferons. There's type 1, which are either alpha or beta, and these are both binding a common receptor. It's a heterodimeric protein on the plasma membrane. So there's interferon alpha there. Then we have interferon gamma, or type 2, which binds a different receptor, and interferon 3 or interferon lambda, which binds a yet different receptor. And the key here is that these interferons are produced, again, by cells in which infection is sensed. The interferons bind cell surface receptors, so they're released from cells, and that initiates signaling to produce the cytokine, the interferon-stimulated genes that are going to inhibit viruses. So the interferons themselves actually do not have antiviral activity per se. So when you make interferon, and you do so rapidly within hours of infection, and then it goes away because you don't want to make interferon continuously, as you'll see in a moment. So here's a cell that's been infected. Its viral RNA is produced. It's sensed in the cytosol. We get transcription of interferon genes. The interferons come out of the cell. They're secreted, and then they will bind to interferon receptors on the cell surface. That initiates another signaling cascade, phosphorylation of proteins and assembly of a transcription factor with STAT1, 2, and IRF9, goes in the nucleus and turns on the promoters for ISGs, interferon-stimulated genes, ISGs, interferon-stimulated genes. There are over 1,000 of those in cells, and they're turned on by the presence of interferon. It can be type 1, type 2, or type 3 interferon, doesn't matter. And the interferon transcription results in mRNAs that are translated to proteins, and the proteins have antiviral activity. Here's one that's breaking up a capsid, apparently, to prevent infection. We actually don't know the mechanisms of action of most ISGs, but I want to tell you a couple that we do know because they're very cool, and how viruses antagonize them, which is also very cool. ISGs. Now, the promoter, this promoter here, that's bound by this transcription factor after interferon is binding the receptor. These promoters come from retroviruses. Amazing. Endogenous retrovirus LTRs regulate the interferon response. So you remember that when a retrovirus infects a cell, if it's a germ cell, it becomes endogenized. And now we have this endogenous retrovirus with the LTRs, GAG-POL envelope. And over time, cells have stolen parts of these endogenous retroviruses. We talked about envelope being stolen for making a placenta. It turns out we've stolen LTRs as well. These have moved around in different places of the genome by recombination, and they drive ISG expression. They are occupying 6 to 14% of the mammalian genome. So I'll go back again. This promoter, responding to interferon, a good part of those have come from retroviral LTRs. And it's not happened just in us, but it's happened in many other animals, as you can see from this phylogenetic tree of different mammals here. They all have captured this LTR, which is an ancient infection, to drive their interferon system. Very cool. Let's talk about the mechanism of action of some of these ISGs. ISGs, interferon-stimulated genes, and the proteins encoded in them. And one of them is a protein called tetherin, or CD137. Tetherin is a great name because it tells you exactly what this protein does. And this protein, as you can see here, is an unusual plasma membrane protein that has two membrane spanning domains. And if you look in the middle here, you can see it's got two transmembrane domains. Then it has a part that's outside of the membrane, then another part that's bound to the membrane. And so what this does, it interferes with virus budding and virus entry. And so here you can see this budding. The virus is not going to come off the cell. It's going to be held on the surface by the tetherin, and big chains of viruses end up, and it will also inhibit entry. So here on the right is a great electron micrograph of a cell infected with HIV-1, in which the viruses are being antagonized by tetherin. They're all stuck at the cell surface, so they can't spread. They make these chains. Beautiful. Now, normally, HIV has no problem with tetherin because it encodes a protein called NEF, which is antagonist. And so there's no tetherin activity in HIV-infected cells unless you take out NEF, and that's what's actually shown in this electron micrograph. So this protein has evolved to block tetherin. It's also similar antagonists are also present in other viruses because tetherin works against a number of envelope viruses. So that's an ISG induced by interferon. That's one of thousands. Another one is called IFIT1. IFIT stands for interferon-induced proteins 1. And that's the actual name of the protein there. And what this does is it binds caps that are not 2'O methylated. So here's a cap structure on mRNA. There's the cap. It's a 5 to 5' linkage, and then that's the first base and the second base of the mRNA. And these cap structures are typically methylated. The cap is methylated, but importantly, the first two bases are typically methylated. And this protein will bind RNAs that lack these 2'O methylations. So here's the figure. Here's an mRNA with a cap. IFIT1 is binding to the cap, and that'll block recruitment of the ribosome because the 4G can't bind the cap if IFIT1 is binding. And so it inhibits translation. On the right are two structures here that show you how this protein binds the cap. Here is a view of the protein where the beta strands are shown as tubes, So all those yellow and green and blue, that's the IFIT1. And then inside is the cap structure. So there's M7G in red. So there's a structure of the two together. So the cap structure is in red. Then the 3' end of the RNA is in there. So it's fitting into a tunnel. So this protein fits right over the cap. Here's a different view where it's a solid view of the protein cut in half. So you can see the cap fitting in this channel. So it's a nice binder of the cap and it blocks translation. But you know what? Viruses get around this. Look at all these ways viruses get around it. First, influenza virus, of course, its caps come from the host cell. It snatches them, right, for priming, and they have 2' O methyls because they're from the cell, and the cell does 2' O methylation, so they're not inhibited by IFIT. Coronaviruses have a protein at the 5' end, right? Because they don't need a cap. They're translated by internal ribosome entry, so they're not antagonized by IFIT1. Some viruses have secondary structure at the 5' end that blocks IFIT1 binding, even if their cap is not methylated, because the protein can't fit there. And then some viral genomes encode methylases, 2' O methylases, that'll methylate the cap, and so the IFIT doesn't recognize it. And some viruses steal host methylases to avoid IFIT1 binding. So you may ask, is this of any use? Well, presumably against some viruses, but these cellular defenses are ancient, and viruses evolve to escape them. They acquire proteins or acquire strategies that will prevent them from binding. It's an arms race. It goes back and forth. The virus changes, the host changes, the virus changes. We'll talk about that later. One more ISG, and I just learned about this one last week and during my visit to Ohio State, the IFIT M3. So we talked about IFIT M1. This is IFIT M3. This one blocks fusion during entry. And it's so cool the way it works. This is a diagram of the protein on the right here and how it fits into a lipid bilayer. So it has a transmembrane alpha helix and then another short segment that fits in between the phospholipid heads, and it's also got some lipid attached to the protein. These red bars are lipids, and it bends the membrane by sticking in there like that. And that's enough to make the membrane not fluid enough to be able to fuse. And so this protein will block fusion, whether it's at the cell surface shown here, or whether it's during endocytosis, that final fusion step where the membrane, the vesicle pinches off, that would be blocked. And here's the mechanism. This is the fusion of virus and host membranes. So we have IFIT M3 stuck in there. It's bending the membrane. And then we have a viral fusion protein, which is in the virus particle. It's attaching to the host membrane, right? You get hairpinning, you get fusion of the inner bilayer, and then the outer one never fuses because this membrane is not fluid enough due to the insertion of IFIT M3. It's very cool. This is apparently very important for influenza virus blocking. All right, so those are just a couple of cool examples. There are thousands of ISGs, and we don't know how most of them work, but each of them probably has a different antiviral role against a different virus and maybe in different parts of the body as well. It's a really complicated system we're just beginning to understand. But what I want to make you understand now is that while the interferon system sounds great, it's actually dangerous also. When you treat patients with interferon, which was done for many years for hepatitis C virus infections, they get very sick because typically in a virus infection, interferon goes on and then off. It induces the ISGs and then goes off. Most cells in the body have interferon receptors. And so when you have a virus infection, most cells can respond, and the ISGs have a lot of course, positive consequences. They can block virus infection, but they have negative ones as well. They cause fever. They cause chills, nausea, malaise, muscle aches. All those things you feel when you have a virus infection are caused by interferons. And every viral infection induces interferon, and that's why we often have flu-like symptoms for different virus infections. Fever, chills, nausea, malaise, flu-like symptoms are not just caused by influenza viruses, but any virus that induces the interferon system. So this is a good example of the pathology caused by an innate immune response. It's not all good. It has to be modulated. Having fever, chills, nausea, malaise, not so bad, but it can be even worse, as you'll see. Our last question, I believe, is, okay, how do interferons limit virus replication? They directly inhibit viral translation. They lyse viral particles. They induce ISGs. They damage cells, or none of the above. All right, how do we do? They induce ISGs. That's right. Interferons don't act directly on viruses. So if you learn that from this, that's important. They induce ISGs, which in turn are the ones that have antiviral effects. And again, we saw some examples of how ISGs do that. The innate system has cytokines, right? That's a big part of it. We talked about interferons. Also comprises sentinel cells, and that is dendritic cells, macrophages, and natural killer cells. And these cells are always traveling through our tissues looking for signs of change, which means something that shouldn't be there, like a foreign antigen, a virus, or something else. So they're in every organ in our body. They're in our blood, in our lymph. They're underneath our skin, everywhere, looking for change. Here's a dendritic cell, a couple of dendritic cells on the left. And these were named dendritic because they had these dendrites, kind of look neuronal, right? And on the right, these are two NK cells that are interrogating this red cell here. And we'll talk about what's going on in both of these. So these are the sentinels in addition to macrophages, which are not here. Now, dendritic cells, as I said, patrol all of our tissues. There are many different kinds of dendritic cells. There are blood dendritic cells. There are tissue dendritic cells. You don't need to be concerned too much. What I want you to remember, though, is that they're always patrolling. Here's an example of dendritic cells in the intestine. Here, this is the layer of epithelial cells here at the top. And viruses are the red ones. They're infecting. And here are dendritic cells that are looking for trouble. These cells are going to be infected. These epithelial cells and the dendritic cells are going to be alerted. This purple one, is pushing its process through the epithelium into the lumen. So it can even sense things in there to see if there's some trouble going on. And there are dendritic cells in all different parts of your body, epithelia right beneath your skin. They're always patrolling and looking for signs of infection, many of them in your body. And when they sense something, then they get activated and they will go into the nearest lymph capillary, make their way to a lymph node, which is shown here. And there they will talk to T cells and B cells. And so here in the T cell zone, we have a number of dendritic cells that have sensed something awry, and they come in here. They're actually presenting antigens and asking the T cells if they're foreign or not. That's a process we'll talk about next time. And on the right is a very cool picture, which shows it's part of a lymph node. And here we have blood capillaries in red. And then the T cells are in blue. They're stained with specific dyes. And then this is a dendritic cell in green. And you can see it's snuggled up to the T lymphocyte. And it's saying, I found this outside there. Should I worry about this or not? And the T cell will tell it, actually. The T cell knows, not the dendritic cell doesn't know. It could be bringing just self-peptide, in which case the T cell would not do anything. But if it's foreign, then things will happen, and that's the subject of next time. So that's what the dendritic cells are doing. That's why we call it a sentinel, because they're patrolling everything, looking for something different in the form of viral proteins and nucleic acids. And that's summarized here again. We have our virus infecting an epithelial layer. We have virus replication, the sensing of infection, production of cytokines. The dendritic cells that are patrolling will come over and sense those cytokines. They're not normally produced, so it will get activated. And as it says here, the dendritic cell can be activated by cytokines produced by infected cells. So that's a specific marker of an infected cell. And also these cells, as they die or they apoptose, they're going to release viral proteins and nucleic acids. And as you'll see in a moment, they have receptors to detect those. And then the dendritic cells become mature. Then they look like a dendritic cell. They enter a lymph capillary. They go to the lymph node. And if this is an infection, the T cell will recognize that. B cells will recognize that. And within a few weeks, you'll have production of antibodies and virus-specific T cells, which will be the subject next time. But this is the link between innate and adaptive defenses. That's why the innate system is so important, because it can sense infection very early. And if it can't control it by the production of cytokines and interferon-stimulated gene products, then the dendritic cells will inform the adaptive response that help is needed. And how do the dendritic cells know? Well, if infection proceeds above a certain level and you have massive amounts of cytokines being produced, that will send many dendritic cells in here and start the adaptive response. So specific cytokines are also involved, and that we'll talk about next time. So here's the progression from immature to mature dendritic cells. So the immature on the left, these are patrolling us, and they have various receptors. They have TLRs, they have cytokine receptors. They can do phagocytosis and take up apoptotic bodies that are coming from dead and dying cells. And they have MHC receptors on their surfaces in the endosomes, and they can endocytose these dead and dying cells and take up the peptides and display them to T cells later on. They can also make interferons. And so if they sense cytokines, if the TLRs sense viral proteins, then these become activated. They migrate to the lymph node, and here again what they look like there. They have now on their surfaces MHC class II molecules, and we'll go into those in more detail next time, but they will be displaying the peptides, these orange bits here. Those are the peptides that were sensed when, say, these dead and dying cells were endocytosed, and the proteins in them are digested and displayed, and they're displayed in MHC specifically for the reason to present them to T cells, and the T cells will decide if this peptide is foreign or not. And this presentation is accompanied by cytokine production by the DC, so the T cell takes all of that into consideration. It takes the peptide and the cytokines into consideration, decides whether this is foreign or not, and if it's foreign, then the T cell gets activated and will go on and become an effector cell and take care of virus-infected cells, as we'll see next time. So that's the activation of dendritic cells. The other sentinel we want to talk about are NK cells, natural killer cells. These are these little brown cells here that scrutinize cells, and they have two receptors that they use that interact with cell surface proteins. They have an inhibitory receptor, which will recognize an MHC molecule on the cell surface. And then they have an activating receptor, which will initiate the killing of the target cell. So if there's no MHC on the surface of the cell, the NK cell activates the killing of the target cell, because many virus infections, as we'll see, downregulate MHC2 from cell surfaces to protect them, and the NK cell is looking for that. They're looking for an absence of MHC molecules, and so here the X means they didn't detect an MHC. Normally, the interaction of the inhibitory receptor with MHC would suppress the activating receptor, so the cell is not killed. So if an MHC is detected by an NK cell, it says, okay, this is not infected, so we will not kill the cell. But no MHC, the cell is killed. And of course, there are viral modulators of NK cells that do all kinds of things, including putting up decoy MHCs to engage the inhibitory receptor so the cell is not killed. That's one of 5 or 6 different strategies that are encoded in virus genomes to avoid NK cell killing. You have to antagonize at every step of this process, otherwise the virus will kill you. Let's talk a little bit now about this concept of inflammation. So we mentioned this before, that a virus infection leads to an inflammatory response. And what I mean is what we have talked about today, the production of cytokines and chemokines, another protein induced by innate responses we haven't mentioned. And these cause inflammation. What is inflammation? Redness, pain, heat, and swelling. This has been known since the 1st century AD. A Roman by the name of Celsus called them rubor, dolor, calor, and tumor. Redness, pain, heat, swelling. Those are the 4 signs of inflammation, which are a consequence of the production of cytokines and chemokines and their biological activities and what they do to cells and tissues. And these 4 signs are caused by cytokines and chemokines that result in increased blood flow. So that is to help get more immune cells to the infected area. Increased capillary permeability. So immune cells can get out of the capillaries into the neighboring tissues that might be infected. And of course, that will cause swelling. Influx of phagocytic cells like macrophages and dendritic cells. So they're all coming in the area, and that's going to cause swelling. And tissue damage. These cytokines and chemokines can damage tissues. They actually are meant to damage infected cells. They also will damage normal tissues. So the signs of inflammation, what we call inflammation, are due to the actions of cytokines and chemokines after infection. Now, you must know that inflammation is not just a product of infection. In fact, this is a guy here I pulled off the internet. He's got inflammation in his head. He's got a headache. He's got joint inflammation. Maybe he just worked out a lot. So exercise, muscle activity can cause inflammation. In fact, many diseases cause inflammation, as shown on this picture here. So if you search inflammation on the web, you get this. But what's missing? They don't have infection. Infection leads to inflammation as well. So it's not specific for virus or bacterial infection, but it's certainly a byproduct of it. But other things do it also. Inflammation. And the mediators of inflammation include cytokines, and there are a couple of different kinds of cytokines. There are pro-inflammatory cytokines. That means that they cause inflammation. And they include IL-1, TNF, IL-6, IL-12, the classic pro-inflammatory cytokines that are going to give you, redness, swelling, heat, pain, and so forth. Then we have anti-inflammatory cytokines that suppress the pro-inflammatory cytokines. There always has to be a balance in the immune response. We make interferon in response to infection. We turn it off eventually, because if you make it all the time, you get very sick. And similarly, these cytokines that cause inflammation, you don't want to have inflammation all the time. When the infection is under control, you make anti-inflammatory cytokines that will suppress it. And then we have chemokines, whose job is to recruit immune cells to the area. And so here again on the lower left is a picture of an infected layer of cells and viruses coming in, infecting that's sensed. The cells are producing cytokines and chemokines. And of course, the cytokines have all those effects that can make this capillary more permeable. And so you get some swelling. That's kind of shown here, inflammation, this area, swelling, cells are coming in. And then you're making chemokines. And the chemokines are going to recruit cells. Here we're recruiting neutrophils from the blood. As we'll see in the next slide, they bind receptors that are induced by the chemokines on the endothelial cells. They roll, and then they squeeze through, and they get into the area where there's an infection. So that is inflammation. Now, these cytokines are soluble. They don't just stay at the site of infection. The cytokines and the chemokines can spread throughout your whole body. So here's a picture of someone with an infection. It could be anywhere, but all these cytokines and chemokines have global effects. So they initially work to help antiviral defenses, but they enter your circulation and they can make you sleepy, lazy, give you muscle pain, loss of appetite, nausea, and many other things. And that's why if you have influenza, which is a lung infection, you can have nausea. It's not because the virus is there. It's because in some people, these cytokines target the gut and make nausea and muscle pain, lethargy. They go in your brain, and they can affect how you're perceiving things as well. So localized infections can have global effects. That's part of inflammation. In fact, some of these cytokines also go into the bone marrow, and they stimulate the production of more lymphocytes. That's one of their jobs. So you don't have enough in your circulation sometimes to deal with infection. So they go in the blood, the bone marrow, and cause more to be made. So that's why you have these global effects. That's why you have flu-like symptoms, because cytokines can't be restricted locally. They diffuse throughout your body. Here's a really interesting diagram to show you how chemokines recruit cells. So cells have receptors for chemokines. So on the left here is a chemokine receptor. The chemokine is in red here. It's binding its receptor. And like many of the other ligand receptor interactions we've talked about today, the binding induces a signaling cascade that here, phosphorylation of a transcription factor that then goes in the nucleus and turns on other genes that will give you a biological response. And what do I mean by that? Well, here on the right, this is a endothelium in an infected area. Chemokines are made by infected cells. They induce on the endothelial membrane a number of receptors that will attract neutrophils. So the neutrophils are barreling through the capillary. They're very close. The capillary isn't very wide, and these occupy much of the space. And then some of these chemokines, cytokines induce the production of chemokines as well as receptors on the surface. And this one will attract the neutrophils to come to the surface. And then there are a series of receptors on the neutrophil which will bind these induced molecules on the endothelium. So these are induced in the area of the infection so that the neutrophils are not just binding everywhere. A neutrophil will bind, it will roll and stop, and then go through the endothelium, can squeeze through. And then it will be in the outside of the blood vessel. It can work on the infected cells in various ways. And of course, if this neutrophil happens to be infected, it'll bring viruses in with it. Of course, virus genomes encode countermeasures to cytokines. And there are 3 different categories shown here. Some viral proteins interrupt the production of cytokines. They can interfere with their synthesis, both cytokines and chemokines. Or they can inhibit the production of functional cytokines. If there are some post-translational modifications are needed, they can be interfered with. The viral proteins can interfere with cytokine action. Some viral genomes encode actually homologs of cytokines, which do not have biological activity, but they will still bind the receptor on the cell surface, but they will not induce a signaling cascade, and they'll block the ability of the real cytokine to bind that receptor. So they're antagonists. And some genomes encode actually soluble cytokine receptors, which are not membrane-bound proteins. They're shed from the cell. They bind cytokines and block them from hitting the real receptor on the cell surface. And then there are viral proteins that alter the signaling. When the cytokine is binding the receptor on the cell surface, if that cell happens to be virus infected, the viral protein can alter the signaling so that the proteins with biological activities are not produced. So a whole host of ways to interfere with this. And I hope you're appreciating now that at every step of innate and we'll see next time intrinsic and innate defenses, there are many, many viral countermeasures, which means that these defenses must be pretty good. So this communication between the innate immune response is very important. It actually stimulates potent immune responses. And in particular, inflammation is key to that. If you suppress inflammation, which again is made during the innate response or occurs during the innate response, you will inhibit potent immune responses. Now, viruses that kill cells, cytopathic viruses, they cause inflammation because the cell is dying, the tissue is being damaged, and viral proteins and nucleic acids are being released, and they can activate the innate response. They can be sensed. They can lead to the production of interferon and cytokines, other cytokines and chemokines, and you get inflammation. These viruses typically encode modulators of innate responses to prevent inflammation, because inflammation leads to a good adaptive response. And some of the viruses we have talked about, adenoviruses, herpes viruses, pox viruses, they all encode antagonists of the proteins that cause inflammation, because that will reduce the adaptive response. Because inflammation is a sign of communication. The dendritic cells responding and the production of cytokines that leads to the recruitment of dendritic cells that accompanies inflammation, that's all meant to activate the adaptive response. So no inflammation, you don't have a good immune response. How do we know this? Well, there are viruses that don't kill cells. Non-cytopathic viruses. They bud from the cell for days at a time and don't cause cell damage. They don't induce apoptosis or necrosis. They probably inhibit it. And so infection is difficult to sense. They may also have antagonists of sensing. So you have a poor innate immune response. You have very little inflammation, and you don't get activation of adaptive immune response. As a consequence, these viruses typically cause persistent infections. They're rarely or inefficiently cleared, and we'll talk specifically about persistent infections later. Mostly, those are the viruses that don't stimulate inflammation. They don't cause cell damage, so they're not well recognized by the adaptive response. So acute infections are efficiently cleared, and the SARS-CoV-2 is an acute infection. It stimulates good innate responses, probably too much, probably an overexuberant response, which contributes to the lung pathology. But you have a very good adaptive response, and you get a good antibody and T-cell response. But persistent infections do not, and as a consequence, they can infect us forever, like the herpes viruses do. And so that's the big lesson from today, that this inflammatory response, heat, swelling, redness, pain, is a reflection of the communication of the innate and adaptive defenses, because it shows that the innate response is well activated. And if it's activated, the dendritic cells are going to communicate with the T and B cells, and you're going to have good immune defenses. If you don't have good inflammation, you typically get an ineffective adaptive response. And a really practical example of this is when you make a non-infectious vaccine. For example, if you take poliovirus and you inactivate it with a chemical to make it non-infectious, it doesn't stimulate a great adaptive response because it's not replicating. So typically non-replicating, non-infectious vaccines, we add adjuvants, which stimulate infection. And we'll talk about those in the vaccine lecture. The adjuvants, the chemicals we add with the vaccine that are injected into you, they stimulate inflammation. If you've ever gotten an inactivated flu vaccine in your shoulder muscle, your deltoid, if it hurts within a few hours, that's inflammation. Heat, swelling, redness, pain, that means it's working. It's good. If it doesn't hurt, it may not be as good. And people have various responses to immunizations in terms of pain, but pain is good at the injection site. And when you put adjuvants in, as in the human papillomavirus vaccine, you get even more pain because those make more inflammation, and it's good. It's good to get it because you want a good adaptive response. You want good antibody and T-cell production. Finally, as I said before, not all inflammation is caused by infection. Exercise and various diseases like diabetes and cancer, they can all cause inflammation. But there's one example of inflammation that I love in the context of a vaccine. You know, when you scratch yourself, the scratch induces inflammation. Here we have an injury in the epidermis of your skin. You've scratched it. The cells sense that damage. They respond by producing cytokines and chemokines, which will cause inflammation, recruitment of immune cells, right? The neutrophils are rolling and moving through the endothelium. You have an influx of fluid from the permeabilized capillaries and so forth, inflammation caused by a scratch. And you get a scratch and you know for the first hours or so it's red and swollen, right? And then it starts to scab up and heal. And it subsides after a while, which is the right thing to do because you're making other cytokines and chemokines that suppress the pro-inflammatory response. Well, it turns out that this inflammation is a important component of smallpox vaccine efficacy. When you get a smallpox vaccine, and unless you're in the military, you're not going to get this anymore. It's given to you in this bifurcated needle shown on the left with 2 sharp points, and there's a drop of vaccine in it. And they take this and they scratch your skin with it multiple times. So they injure you. And the virus ends up better infecting the epidermis because it wouldn't do well on the dead cells, right? But there's also inflammation caused by the scratch. And that amplifies the immune response because it pulls in all these cells, including dendritic cells, and that makes for a better adaptive response. And in fact, in animal models, if you deliver the vaccine instead of scratching by just injecting it, you get a poorer antibody response. So this is really interesting that this damage caused by the scratching, and this is specific for smallpox vaccine. We don't do this for any other vaccine because the virus likes to reproduce in this skin here, is really helping vaccine efficacy. Finally, last thing I want to say is an emphasis of everything I have told you. All viruses have to encode at least one regulator of these intrinsic and innate defenses that I've talked about. That includes sensing, interferon production, signaling after interferon binding to receptors, the actions of cytokines and chemokines, NK cells, DCs. They're also antagonists, which we'll talk about next time when we talk about antigen presentation. And the other component that bridges the innate and adaptive responses, which we didn't talk about, which is complement, a series of proteins in the blood that help to detect when there's something that shouldn't be there. So now we are ready to talk about adaptive responses. We'll do that next time. And what we're going to talk about is how these activated dendritic cells, activated by infection, what they actually do in the lymph node to stimulate the production of antibodies that are going to block virus infection and cytotoxic T lymphocytes that will kill virus-infected cells. And so next time we will talk about adaptive immunity. However, of course, you have next week off. So to all of you, I wish you a really good week off and look forward to that Monday when we all come back virtually again. Unfortunately, we're not going to see each other, but I will have office hours at the usual time via Zoom if you want to ask questions. And if you can't make office hours, you can schedule a Zoom with me because now you don't have to schlep up here to the medical center. We can just do it via Zoom. So, and the next exam will be again online, although hopefully it'll be better. So again, thank you very much and have a great vacation. See you next time.